Hi, my name is Tanvi, and today I'm going to be talking about gene regulation in bacteria. Have you ever wondered how bacteria control which genes are turned on or off? Well, that's where operons come in. Today we'll be modeling two important operons separately, the lac operon and the TRP operon. Let's dive in. I'm first going to go over some background questions before I get into deeper detail about how each operon functions. However, I'll be discussing questions 5 and 6 deeper in the video with a visual model. So what is a gene? A gene is a segment of DNA that contains the instructions to produce a specific protein or RNA molecule. Genes are the functional units of heredity, and they play a central role in determining traits in all living organisms. What is the role of RNA polymerase in protein synthesis? RNA polymerase is an enzyme responsible for the transcribing of DNA into RNA. It binds to the promoter region of a gene and synthesizes a complementary RNA strand from the DNA template, which is later processed to produce proteins. What is the role of ribosomes in protein synthesis? Ribosomes are the cellular machinery that translate mRNA into proteins. They read the mRNA sequence, match it with the correct tRNA carrying amino acids, and link the amino acids together to form a protein. So what are the roles of an operator, promoter, and structural genes within an operon? A promoter is a DNA sequence where RNA polymerase binds to initiate transcription. An operator is a DNA sequence that acts like a switch controlling access of RNA polymerase to the structural genes. It's the site where repressors or activators bind. Now, structural genes are the code for proteins or enzymes needed for specific cellular functions, such as breaking down lactose in the lac operon. So, what is a regulator gene? A regulator gene is a gene that encodes a protein, such as a repressor or activator, which controls the expression of other genes in an operon. It regulates whether the operon is on or off. So starting off with the lac operon. The lac operon controls the breakdown of lactose in bacteria. It consists of several key parts: the promoter, the operator, the regulatory gene, structural genes, RNA polymerase, and the repressor protein, all of which I discussed a couple minutes ago. In its default state, when the lac operon is turned off, the regulatory gene produces a repressor protein, which binds to the operator and blocks RNA polymerase from transcribing the genes that break down lactose. However, when lactose is present, it acts as an inducer by binding to the repressor. This changes the repressor's shape, making it detach from the operator. Now, RNA polymerase can proceed with transcription, producing enzymes that digest lactose. So, in summary, the lac operon is normally off, but gets turned on in the presence of lactose. This helps bacteria only produce lactose-digesting enzymes when needed. Now let's take a look at the TRP operon, which controls tryptophan production. Unlike the lac operon, this one's usually on because bacteria constantly need tryptophan to build proteins. In the absence of tryptophan, the repressor protein is inactive. RNA polymerase binds to the promoter and transcription proceeds. This produces enzymes that synthesize tryptophan. When tryptophan levels are high, tryptophan itself acts as a co-repressor by binding to the repressor protein. This activates the repressor, which then binds to the operator, blocking RNA polymerase and stopping transcription. So, in all, the TRP operon is usually on but gets turned off when tryptophan is abundant. This prevents unnecessary tryptophan production. I'm now going to dive into analysis and conclusion questions, where I'm essentially going to be reiterating everything I said earlier in the video. So I'm first going to describe what takes place when each operon is activated and inactivated. So when either the lac operon has lactose present or the TRP operon doesn't have any tryptophan, the repressor protein is either removed from the operator or inactive. RNA polymerase binds to the promoter and transcribes the structural genes into mRNA, which is translated into proteins. Now in inactivation, when the operon is inactivated, in the lac operon, this is without lactose, and in the TRP operon, this is with tryptophan present. So in this, the repressor binds to the operator, blocking RNA polymerase, and transcription of the structural genes stop and no enzymes are produced. So what substance serves as the repressor in the TRP operon, and what serves as the activator in the lac operon? So the repressor in the TRP operon is the repressor protein, which is activated by the co-repressor tryptophan. And in the lac operon, it's lactose, which acts as an inducer by binding to the repressor and preventing it from blocking transcription. So how might a mutation in the regulator gene affect the operon? So a mutation in the regulator gene could disrupt the production or function of the repressor protein. For example, if the repressor is non-functional, the operon may permanently remain on, leading to unnecessary protein production. If the repressor is constantly active, the operon may remain permanently off, preventing the production of necessary enzymes. So how would the loss of the promoter site affect the production of enzymes by the operon? So if the promoter site is lost, RNA polymerase would be unable to bind to the DNA and initiate transcription. As a result, the structural genes would not be transcribed and no enzymes would be produced, regardless of environmental conditions. So how is the function of an operon similar to feedback inhibition? So both operons and feedback inhibition regulate processes to conserve energy and resources. In operons, the presence or absence of a molecule, in this case is lactose and tryptophan, they both determine whether genes are transcribed. In feedback inhibition, the end product of a metabolic pathway inhibits an enzyme earlier in the pathway, preventing overproduction. Both mechanisms ensure the cell only produces what it needs when it needs it. So, to recap, the LAC operon is normally turned off and turns on when lactose is available, while the TRP operon is normally on and turns off when tryptophan is abundant. These regulatory mechanisms essentially help bacteria efficiently manage their resources. Thank you so much for watching. I hope you enjoyed this video.